Sure.
Good morning, dear ladies and gentlemen. We extend a warm and a pleasant welcome to everyone on this cheerful morning. We are pleased to welcome the staff, the students from various colleges. We had a delightful and enlightening experience yesterday listening to the talks of the experts. Likewise, let us enrich our knowledge from today's technical session too. We extend a very warm welcome to Dr. Arjun Masarkar sir, who will be taking on the technical session 5. And to introduce Dr. Arjun Masarkar, we are having our head of the department, Dr. G. Nidhirajan sir. I'm handing over the mic to Dr. Nidhirajan sir for the introduction and for chairing the session too.
One and all present here. I am Nidhirajan, head of the department, PG and Research Center in Biotechnology, MGR College, Hosur, India. I am honored to introduce Arjun Vijay Masarkar, MD, PhD, in the International Conference on the Genetic Disorders in Human. Professor Arjun Vijay Masarkar did his undergraduate degree in electrical engineering and MD in medicine and PhD in neuroscience. And he did a postdoctoral fellow in Columbia University, and he has a lot of certification, license, and he has appointed many various positions like instructor and co-director, director, investigator, and principal investigator, and he has got many funds from various funding agencies, and he got many awards and honors, and he occupied many positions like director, etc. He has published more than 32 research articles in peer-reviewed journal, more than 10 books and reviews, more than eight papers in under review process, and manuscript in preparation is about 10 articles. He has submitted 61 abstracts in various conferences and symposium. With this brief intro, I hand over the presentation to Professor Arjun Vijay Masarkar. Thank you, sir.
Well, thank you very much for that very nice introduction. Can everyone hear me?
Yes, yes.
Yeah, okay, great. And I will go ahead and share my screen. Oh, I see. Just do this here. Okay, can everyone see the PowerPoint? Yeah. Great.
Yeah, I can see.
Okay, so I'm really happy to talk to you all today about my talk titled "Towards a Molecular and Cellular Epidemiology of Early Alzheimer's Disease." And I have no disclosures. And first I wanted to acknowledge the hard work of my team. I have both a basic science and a clinical laboratory at NYU, and I have a lot of colleagues that I collaborate with as well. I'm going to really be highlighting the work of Isabel Reyes and Chengju Tian in my lab. And of course, Mohan Kumar Thangavel has also joined my lab last year, and he's on the call as well. So very happy to have him at NYU in my lab. So the agenda of my talk is broken down as follows. First, I want to give you all a brief primer on what is Alzheimer's disease so that we're all on the same page. Then I'm going to talk about Alzheimer's disease susceptibility at a macro scale, a large scale, really focusing on the entorhinal cortex and area CA1 of hippocampus as vulnerable regions. And then I'll talk about really honing in on the area CA1 of hippocampus, discussing the functional heterogeneity of subpopulation of neurons in that area. I'll then talk about the differential effect of Alzheimer's disease on those subpopulations in experiments that we've done in transgenic models of AD. And lastly, I'll end with some data on the differential and molecular neuropathology of CA1 subpopulations in human Alzheimer's disease. And for those who don't know the pictures that I'm showing to the right, in the upper left picture is Dr. Alzheimer, for whom the disease is named. He was the first person to really describe the pathology of Alzheimer's disease, how it's defined today. And the first patient in whom he described it in is depicted on the right. And below is a cartoon of the hallmark pathologies of Alzheimer's disease, the sort of cottony looking amyloid plaque below, and the tau-based tangle, sort of the dark triangle appearing neuron above that. So first, a brief primer on Alzheimer's disease. We talk about the word Alzheimer's, we talk about the word dementia. What does it mean? I think we have to really contextualize for the rest of the talk. So dementia is a loss of cognition or behavioral abilities that were previously acquired to the degree that one loses their independence. They're not able to perform normally in their daily life, social functioning, or at their occupation. The pathway to dementia is a series of stages going from normal to end stage. And it's a progression. So you have normal at the top, preclinical, meaning there are symptoms or biological signs. So there are no symptoms, but there's some biological signs. We then have a very important, yeah. Okay, sure. Presentation mode. The presentation mode. Yeah. Yeah, where do I get the presentation mode? Yep. A couple like symbol, yeah. Yeah, that's what I clicked. Do you see that?
Yeah, able to see mobile.
Ah, okay. I mean, right now, do you see kind of like the minimized screen? Sure. Yeah, I can do that. Sorry about that. So I'll do the stop presenting. And then what I'll do is, I guess I'll open up my presentation first. Maybe, then I'll go to my slideshow. Let's see. No. Show presenter view. Maybe that's what I need. Maybe that's what it is. So what we'll do is, I think I got it. Window. We'll do PowerPoint. Allow. And then we'll go here. And then we'll do this one. Is that better?
Can you hear?
Yeah.
Yeah, is it fine? Yeah, I can see in my mobile his presentation slide also I can see. Okay.
Huh, it's actually in the presentation mode. Oh, okay. That's okay. At least I'm not the only one. Select slideshow tab, and then, okay. Maybe this is what I should do. How about now? Now? Uh-huh. Oh, you know what? I accidentally stopped the sharing. But I think I know, I think I'm supposed to hit the presenter view in the slideshow menu of the PowerPoint software. Then I can share either a window or my screen, but I'll take a window. Okay. How about now? And then how about this? Does this work? F4? No. Ah, Shift F5. Okay, I did that. Oh, how about this? How about this? You know what? There's another way that I can do this too. If so I can either I can actually share my entire screen. So if I share my entire screen, I think that should work. Let's try that. Share my entire screen. Let me just make sure yeah, entire screen. So you'll see this, but then I will then take over the whole screen with the PowerPoint. How about that? Okay, all right, we got it. Okay, on the fly. Okay. So where were we? Yeah, so dementia really is a spectrum going from normal to end stage. And so there's a critical stage in between called mild cognitive impairment, where there is a little bit of objective cognitive deficits, but the person is relatively intact with their activities of daily living. And then so on and so forth. You get the dementia stages after that, until there's very little functioning in day-to-day life and at home. Now, the causes of dementia are quite broad, and Alzheimer's disease is the most common, which is why you hear about it the most. It's about 60 to 70% of all of dementia cases. What is Alzheimer's disease biologically? It's defined by its neuropathology. And in essence, there's two main pathologic proteins, amyloid plaque and tau-based neurofibrillary tangles. The amyloid plaque is outside of the brain cell, and the tau-based tangles are inside of the neurons. Now, the problem is that Alzheimer's disease prevalence is growing at a very rapid rate, as you can see from the chart here. And so this is a huge problem, in terms of burden on society, financially, and also a lot of burden on the caregivers. And so we really need new treatments to slow Alzheimer's disease progression. So what do we know about Alzheimer's disease? Well, this graph sort of encapsulates what we know about human Alzheimer's disease. We know that there's a series of processes which we think begins with the rise of amyloid. And this rise in amyloid may even begin in our 50s, as you can see by the leftmost red curve. After that, you get elevation in tau, then you get destruction of brain structure, followed by memory loss and followed by loss of clinical function. Now, in the beginning, a lot of the trials had focused on dementia stage and also the MCI stage, but a lot of those trials have failed. And so now the scientific focus has really been shifted towards the impairment, preclinical and cognitively normal stages of the disease. And so we really need to understand the disease at these early stages. So in that context, I will talk about susceptibility to Alzheimer's disease in these early stages. So MRI technology had allowed us during the 2000s to really establish that the medial temporal lobe is involved early in the disease. And so as indicated here by this coronal MRI section, the hippocampi and the parahippocampal gyrus, which are indicated by the arrows, become smaller in Alzheimer's disease, especially early on. And the smaller those volumes are, the more likely the person is to progress. So this seems to be a critical node of early Alzheimer's disease. Now, what does it correspond to neuropathologically? So there's this staging of Alzheimer's disease called the Braak stage, and it goes from 1 to 6. Now, loosely, normal people are going to be Braak stage 0, where there's very minimal or no pathology in the brain at all. People who are mild
Students,
cognitive
can you please
impairment,
settle
who have
down?
pre-dementia, basically,
The talk is going
are
on.
going
Kindly
to
please
be
settle
Braak
down.
stage 1, 2, or 3.
Please
And
don't
that's
disturb
where
others.
the tau
Settle down.
pathology is really restricted to the entorhinal cortex and the hippocampus. Once you start to get to Braak stage 5 and 6, these are largely going to be people with dementia stage of Alzheimer's disease. So when we're talking about early stage Alzheimer's, we're really focused on stage 1 through 3, where we are really referring to entorhinal cortex involvement and hippocampal involvement. But even within hippocampus, there's a susceptibility, and that's the CA1 area. So to the left is depicted the hippocampal formation in human, and as you can see that there are 5 domains of the hippocampus: cebiculum, CA1, CA2, CA3, and CA4. CA1 is the main output area of the hippocampus, and it seems to be the part of the hippocampus that's affected the most early in the disease and most robustly by the pathology. And this is evidenced by the panels that I've shown here. And so you can kind of look back at that Braak staging and more precisely indicate that this sort of mild cognitive impairment stage probably corresponds to the transition of pathology from entorhinal cortex to CA1. So that's a critical gateway in this disease. And then it moves from CA1 and beyond. So we really need to get a handle of what's going on within area CA1. Now, when you look at a microscopic level at area CA1, you will see that not all the neurons are exhibiting pathology. So here's an example of a brain from a 72-year-old man with Alzheimer's disease, focusing in on the neurons within hippocampal CA1. And what you can see is that there's a tangle in one neuron, but the neuron right next to it does not. And so why does this happen? Why does one neuron have the pathology? Why is it affected? And why is the other neuron not? And so what are the rules that would really dictate why this happens? And so I kind of look at it through an epidemiology lens to your dysfunction due to Alzheimer's disease or the pathology that may arise. And what does it have to do with who you are, your intrinsic physiologic properties or your genetic molecular properties? And so this is the lens through which I conduct my research in Alzheimer's disease. And so owing to this framework, I wanted to speak to the functional heterogeneity of CA1 subpopulations. And so just to preface that, I wanted to review briefly the canonical neural circuit of area CA1. So what I've depicted here is a pyramidal neuron of CA1, the main neurons, and it receives input from various areas in the hippocampus and outside. So the entorhinal cortex gives inputs to the distal dendrite of the neuron, and in that same location, the thalamic inputs from nucleus reunions of thalamus come in as well. Area CA3 of hippocampus relays dentate gyrus input at the proximal dendrite, and the basal dendrite receives information from area CA2. Now, all of this information, other than the thalamus, is routed through entorhinal cortex. So entorhinal cortex provides inputs both directly and indirectly via dentate gyrus and CA3 and CA2 to area CA1. So you have direct pathway and indirect pathway. There are multiple anatomical axes to the area CA1 of hippocampus. So one axis is called transverse, that's going from subiculum to CA2. One axis is called longitudinal, which is in the mouse dorsal to ventral. In human, it's called posterior to anterior. And then you have the radial axis, which is the depth axis. How deep in the brain is the neuron? And so there have been a lot of studies, including from my lab, in how does the functioning and the properties and the genetics of these neurons vary across these anatomical axes. And what we found is that there's more spatial information in the proximal side of CA1 when we're looking at the transverse axis towards CA2. And so non-spatial information, sort of olfactory responses, for example, are found more in distal CA1, which is towards subiculum. There's a hypothesis that the transverse axis function, the sort of dichotomy between spatial and non-spatial function, derives from the entorhinal cortex input that comes to those regions. And so it's hypothesized that proximal CA1 is driven by the spatial part of entorhinal cortex, or medial EC, and whereas the distal part of CA1 is driven by the lateral EC, which is specialized for non-spatial inputs. And so what we did is we used mouse in vitro slice physiology and optogenetics to test that hypothesis. And what we did was we expressed the light-sensitive ion channel rhodopsin 2 in axons of either medial or lateral entorhinal cortex. And then once we made our in vitro prep, we recorded intracellularly from neurons in proximal or distal CA1 and used light to stimulate the MEC or LEC inputs and recorded the responses. And what we found was quite striking. Indeed, the medial entorhinal cortex gives very strong excitation to proximal CA1, but very minimally to distal CA1. Whereas lateral entorhinal cortex did the opposite. It gave a very powerful excitatory input to distal CA1, but a very small input to proximal CA1. So this basically proved the hypothesis that both anatomical and functional studies had put forward. There's also the radial axis I mentioned. Now the radial axis is also quite interesting because there are molecular studies that show that these layers are distinct, as you can see from the listing of genes that are differentially expressed in these two layers. But there's also evidence that these two layers are functionally distinct. So similarly, spatial information seems to be more heavily expressed not only in proximal CA1, but in the deep layer, whereas non-spatial responses, olfactory, for example, features of objects, are found more in the superficial layer. And so again, similar to the transverse axis, we hypothesized that radial axis function may derive from the character of its efferent input. And so using a similar optogenetic strategy, we now recorded from deep and superficial cells with channel rhodopsin expressed either in medial or lateral entorhinal cortex. And what we found to the right is that medial entorhinal cortex preferred to excite deep neurons, preferentially in proximal CA1, of course, and lateral entorhinal cortex preferentially excited superficial layer neurons in distal CA1. So not only did the inputs determine the proximal distal functional difference, but they also dictated the deep superficial difference. Us and other groups have also found that the deep layer receives more synaptic inhibition. So as some of you may know, the neurons can either be excitatory or inhibitory in the brain. And so interneurons, specifically parvalbumin interneurons, seem to favor deep neurons, the deep pyramidal neurons. There are other interneurons that are more associated with superficial cells, superficial neurons. In addition, the superficial neurons appear to inhibit deep neurons via those same PV interneurons. And thus, what we and others find is that the deep neurons actually display lower excitation to inhibition ratios. When it comes to intrinsic properties, intrinsic excitability, the deep neurons actually show an enhanced intrinsic excitability. So their ability to fire action potentials, which are the main currency in the brain, are much higher than superficial neurons, both in proximal and distal CA1. And this seemed to be related to basic properties of their intrinsic firing. And our data sort of suggests that there may be certain ion channels that may be differentially expressed to give rise to that increased excitability. We've also found, similar to other groups, that deep neurons show more activity in vivo. So it appears that although that they may have more inhibition synaptically, the enhanced intrinsic firing wins out. And so we've used miniscope imaging, which essentially means these are implantable microendoscopes that can pick up fluorescent activity in the brain. And so we can express through an AAV vector the fluorescent calcium sensor GCAM within CA1, and then use the miniscope cameras to pick up the fluorescence, which is representative of calcium activity in neurons of interest during a behavior. And so what we found is that when a mouse is moving along a track, it's the deep layer neurons that show more activity and more firing, and more firing specifically in accordance with spatial exploration. And so this again shows that it's actually the intrinsic excitability of the deep neurons that win out over the differences in synaptic excitation. So now that we had been armed with this physiologic information and connectivity information, we then pursued work in Alzheimer's disease models. So we used the triple transgenic Alzheimer's disease model, which expresses both amyloid and tau. And so we performed slice physiology recordings at an age where there should be both pathologies. And what we found was that in terms of intrinsic excitability, in these triple transgenic mice, firing rates were reduced in the superficial neurons, and they were increased in deep neurons. And we find that there's this slow after hyperpolarization. So the suppressive potential that is typically associated with aging was preferentially enhanced in the superficial layer as opposed to the deep. And so what this shows is that Alzheimer's disease pathology, regardless of where it is, appears to impact the superficial layer more than the deep layer in terms of inhibiting their intrinsic ability to fire. And in almost a compensatory way, the deep neurons are hyperactivated. When it comes to inhibition, we also see changes that promote excitability of the deep neurons. So as I mentioned, deep neurons are preferentially receiving inhibition. These same interneurons are actually affected by amyloid in a way that would suppress their activity. So if there's less inhibition, there's more net excitation of these deep neurons. And this would hypothesize that the excitability of deep neurons is even more enhanced. When it comes to excitation, excuse me, we focused primarily on the entorhinal cortex excitatory inputs. And what we found in our patch lamp recordings was that there was a selective suppression of entorhinal cortical excitation onto superficial cells compared to the deep neurons. The deep neurons were essentially unchanged, as shown in the upper right panel. And we recapitulated this same data using not only electrical stimulation, but also our optogenic paradigm, where we selectively stimulated lateral and medial entorhinal cortex inputs in both distal and proximal CA1. So it didn't matter where along the hippocampus, the CA1 transverse axis the neuron was, the superficial layers seemed to be more vulnerable to loss of input, and the deep neurons were more protected. And the mechanism we find appears to be both loss of spines, so it's a postsynaptic effect that's spine dependent, but there's also a postsynaptic spine independent mechanism as well. And we found this to be true both in older triple transgenic mice, younger triple transgenic mice where there's only amyloid, as well as the 5XFAD model, which has only amyloid. And so this appears to be an effect that's really stimulated by amyloid alone. When it came to the indirect inputs of entorhinal cortex, the CA3 inputs, we found a similar selective vulnerability that also appears to be amyloid driven, but this effect was primarily spine loss independent. So in sum, these data show that intrinsic vulnerability is a hallmark of superficial neurons, and that deep neurons are resilient to the physiologic and synaptic destructive impact of Alzheimer's disease. I was also interested in going back to some of the genetic differences between these two layers to see if there are any clues underlying this differential susceptibility. And lo and behold, 2 of the genes have been well studied in relation to Alzheimer's disease effects. For example, the serotonin receptor gene, HT1RA, has actually been found to promote amyloid-induced tau phosphorylation. Furthermore, there seems to be a differential expression of the CGN kinase in the superficial layer. And this kinase has been shown to facilitate tau phosphorylation and amyloid-induced spine loss. And so there actually is a molecular genetic mechanism, perhaps, for the differential susceptibility that we're seeing. Now, that's all said and well in the mouse models, but what about human? We always have to recapitulate or affirm our findings in human AD. So it's quite interesting. So I had shown before that the lateral entorhinal cortex appears to primarily innervate or excite distal CA1, and medial entorhinal cortex primarily innervates, excites proximal CA1. And lo and behold, studies that have actually paid attention to this anatomy have actually found that lateral entorhinal cortex is affected before medial entorhinal cortex in terms of AD pathology. And following suit, distal CA1 is affected before proximal CA1. And so this connectivity appears to mirror disease. And this explains some functional changes that we see in patients. We usually see these non-spatial, for example, olfactory deficits before classical declarative memory deficits. So LEC distal CA1 circuit dysfunction before MEC proximal CA1 circuit dysfunction. This type of relationship has also stimulated thoughts about synaptic spread of pathology, but since synaptic connectivity seems to be so important. However, I wasn't completely accepting of this hypothesis because I think that there's a lot of evidence for intrinsic vulnerability. So one problem is the lateral entorhinal cortex also targets dentate gyrus, but dentate gyrus, as well as CA2 and CA3, but these 3 regions are not affected until later. So how could it be that connectivity alone dictates vulnerability? It has to be at least suggesting a strong involvement or co-involvement of intrinsic factors. So what we did was we utilized the NYU Brain Bank and looked at Alzheimer's cases and quantified the degree of amyloid pathology and tau pathology in these two layers, as well as in proximal and distal CA1. And what we found was that, yes, and these were advanced cases. These are Brock stage 5, 6 cases. And what we found is that, yes, there's a slight elevation of pathology in distal CA1 versus proximal, so LEC versus MEC innervation. But really, the gradient wasn't that high. Secondly, when it came to deep superficial, there was a significant increase in pathology in the superficial layer of both amyloid and tau pathology, whether you were in proximal CA1 or distal CA1. And so this actually suggests that connectivity, meaning who you talk to, who you receive input from, is not the primary mechanism, but that intrinsic vulnerability is more important. We then wanted molecular evidence of this hypothesis. And so we turned to a proteomic method. So we are able to use a technique where we can take fixed human AD brains, and we use Brock stage 6, and we can use a specialized microscope called a laser capture microdissection microscope. And we can actually cut out the superficial layer and the deep layer. And then we can use label-free quantitative liquid chromatography mass spec to look at the entire protein ensemble expressed in those layers, and then compare the expression of proteins in one population versus the other. So this method was quite fruitful. We were able to quantify over 3,000 proteins spanning 6 orders of magnitude in abundance. And we are actually able to capture a lot of excitability-related proteins, over 15%. Remarkably, even at an advanced stage of Alzheimer's disease, we found that deep superficial layer differences were still evident. And in fact, one marker, alpha internexin, which was increased in the deep layer, has been shown to be a marker of late as opposed to early vulnerability. And so this actually supported our hypothesis that superficial neurons are affected, may degenerate, develop pathology earlier in the disease, and the deep layers are more resilient and develop pathology, gain dysfunction later in the disease. And so just to conclude my talk, my conclusions are as followed. 1, hippocampal CA1 is affected early in AD, but not all neurons are equally affected. 2, CA1 neurons are intrinsically, functionally, and synaptically heterogeneous across the distal proximal axis and the deep superficial axis. Distal CA1 and superficial neurons therein are driven by lateral internal cortex, whereas proximal CA1 deep neurons are driven by medial internal cortex, giving rise to non-spatial versus spatial functional specialization. Within CA1, Alzheimer's disease leads to hypoactivity of the superficial layer and hyperactivity of the deep layer, regardless of transverse axis position. And then within CA1, AD pathology and related molecular signatures of disease indicate an intrinsic vulnerability of the superficial layer. And these data in total suggest that intrinsic more than extrinsic factors play a role in superficial neuron vulnerability, keying in, owing to the theme of this conference, on molecular genetic factors. And so I'll stop there, and I'm happy to take any questions. Sure. So this will be in the chat. Anybody have questions? Good question. So the question is, as people age, they get memory loss. At what point of time should they visit a doctor for Alzheimer's disease or other diagnoses? That's an excellent question. So I see patients in my practice at all levels. I see a lot of people who are in their 50s, 60s that are perfectly fine, but they're a little bit worried because their mother or father had Alzheimer's and they're worried. So I think it's fine to see a doctor at any point. Now, when do we become really worried? Well, if you're starting to make mistakes every day, that especially noticeable to colleagues or family, then I think for sure it's time to see a doctor. But there's a lot that we can do and modify, even if there aren't so many noticeable symptoms. And I often view myself not necessarily as just diagnosing dementia or Alzheimer's disease, but also helping people optimize their brain health, even in the absence of these diagnoses. Mm-hmm. Great question. So, sir, as you said, only few neurons are tangled, others are normal. Is there a way to untangle the troubled ones? So I'm going to answer this two ways. One is we don't know if the other ones are really normal. Because as you saw, I think it's the superficial ones that become tangled. But the deep neurons are compensating in a way. They are changing. And I don't know if that's a good thing. So one could ask, apart from treating the tangle, should we also treat the other neurons that are not tangled, but maybe not working properly? Now, when it comes to untangling, there have been some strategies out there that have been tried in trials that have been successful. For example, using an antibody or using an agent that can prevent the formation of the tangle itself, for formation of the three-dimensional tangle. They haven't really been successful. I think tau is even harder than amyloid to treat because it's intracellular. I would say that maybe an avenue is to intervene upon the phosphorylation of the tau, because we know that the dominoes start to fall once the tau is phosphorylated in an inappropriate way. So I think intervening upon that process is one way before it becomes a tangle. That's okay. So is the number of tangled neurons related to the severity of the disease? Excellent question. And just to preface this, right, so there's two pathologies, amyloid and the tau pathology, the plaques and the tangles. And they both have stages. One is called the Thal stage, one is called the Brock stage, was what I showed. The Thal stage, where and how many, doesn't seem to indicate the symptom of the patient. But the where and how many of the tangles does. So to answer this question, yes, it's actually the tau tangles that determine the severity, meaning where they are, meaning the location would tell you which symptom, and how many there are would tell you how bad the symptom is. Thank you so much. Well, thank you for the invitation. This is a pleasure, and sorry for the technical issue in the beginning. Okay, take care, everybody. Bye-bye. And good night. Yes. Bye-bye.
Which I'm really proud of. It's the National Health Service. So National Health Service in the England, it's very, very unique. Unique in the sense it is free for all at the point of use. And every resident in the UK, they don't have to pay. It's free of charge at the point of use. So that's what makes NHS so unique, and everybody who's part of NHS feels so proud about the whole organization. So, so what is NHS? So next slide, please, Kirti. So NHS is it starts off with two prongs. You have primary care trusts and the acute trusts. And then we come together under this strategic health authorities where all the policies and the standards and everything is set. Then obviously that's under Department of Health, and Department of Health, like every other country, is under Parliament. So the politicians decide what is good for the patients, not people like you and me. So that's pretty much like every country. But this is the general structure of National Health Service. Next slide, please, Kirti. So this, when Kirti asked me, about the topic, I would like to share an experience that I had a good while ago, maybe 10, 12 years. It was quite emotional, and it stayed and it will stay with me forever. Like Kirti said, I'm a clinician, so it was like everybody, one fine Monday morning, I was there, sat in my clinic when I saw say about early lady, and I know her well, I know her family, I know her son and her husband. So as luck would have it, this lady came to see me completely broken. So normally a general practitioner's consulting time is about 10 minutes, but this consultation went a bit longer. And out of that 10 minutes first, she just cried her eyes out for the first 5 to 7 minutes. There was no way I had to give her the time. There was no way I could console her and just start the consultation. The reason behind that was two people. She had the only son, and then obviously her husband. Her husband, I did a 2-week referral a few months back, and I had sent him for tests, and he was diagnosed with colonic cancer. Then a 31-year-old only son came to see me with a skin lesion. And I just, really, I still remember how I felt that moment when I looked at that rash. It was quite obvious, even clinically, that it was malignant melanoma. So here we have a lady with two people, probably her whole world, both with cancer. Sadly, very sadly, over a period of time, I'm talking about within, say, 12 to 15 months, she lost both of them. So that's the sad story, and that is real life that we come across on a daily basis. So when Kirti came to me asking about the topic, and we were discussing, and I thought, right, I asked about the audience, and she said, you know, it is my students, my colleagues. And I thought this topic will be really fitting, because I really wanted you guys to know that the global community, the patient population, they actually need you. They need your work. So I thought I should bring the story to you guys on behalf of the patients that we clinicians see on a daily basis. So that's why I wanted to choose this. I chose this topic, and I wanted to talk to you guys about it. Sorry about this. I've got a frog in my throat. So, genetics and cancer. So, WHO report says that cancer killed nearly 10 million people in 2020. Remember, we were right in the thick of the pandemic, and the cancer care pathways were a little bit functioning out of norm. And we know that cancer is the leading cause of death globally. And in the West, the cancer risk to general population is 1 in 3 and quickly moving to 1 in 2. So people will, at some point in their life, 1 in 2 people will get cancer. And in the UK, cancer causes more than, sorry about this, 1 in 4 of all the deaths in the UK. This, we don't have an 2021-2022 data, but this is what I'm talking about, the statistics, it's all up until 2019. So there are around 167,000 cancer deaths in the UK every year. We are talking about really 460 every day. That's a big number. And we all know, you know, United Kingdom, in comparison to India, such a big country, what's the size of UK?
Dear students,
so even
please
in
listen.
This country,
She's talking something
related
to your subject.
The size of the
Why
country
your work
into
is important in
if
world.
we look
All
at
that she's
that
speaking,
number,
kindly
that's
listen.
quite staggering. And every 4 minutes that someone in the UK dies from cancer. I don't know how you guys feel when you see these numbers. I was completely flabbergasted, because you don't go looking for the statistics on a daily basis when you're just, so busy with other things. But when I just started to prepare for this presentation, that's when I actually knew that every 4 minutes, someone in the UK is dying from cancer. And that's really sad. We are supposed to be really advanced, with all the work that you guys do to help with patient care. So it highlighted that there's more work to be done. Almost half of all cancer deaths are lung, bowel, breast, or prostate cancer. Again, this is just keeping in with UK in mind. Almost half of all cancer in males are from lung, prostate, or bowel cancer, and in females, it is lung, breast, or bowel cancer. So you can see it's the lung and bowel are the commonest cancer in both sexes. Please, Kirti. Having said all this, UK mortality is ranked lower than 2/3 of Europe. And that's saying something, isn't it? Yeah. So let's talk about the common cancers that we normally see in the UK. We did say that the 4 common cancers are lung, breast, bowel, and prostate. Now, if we look at lung cancer, it is the 3rd most common cancer in the UK, and it accounts to about 13% of all new cancer cases. And we know that all the smoking cessation advice and everything has been there only in the last 2 decades. But in the 1960s, smoking, it was it was some sort of a fad, wasn't it? So, but we are actually seeing the consequences of that now. And we also had all this asbestosis and the mining and everything that just contributes to it. So there are around 48,500 new lung cancer cases in the UK every year. And lung cancer, and there are around this again, for me, the mortality is what it's just really important to actually highlight the amount of work that needs to be done. There are around 34,800 lung cancer deaths in the UK every year. And looking at the breast cancer, it is the most common cancer in the UK. And there are about 55,900 new breast cancer cases every year. And if you look at the number of deaths, it's 11,500 every year. If you compare that with the lung cancer deaths, 34,800 to 11,500, you might wonder, how is it that it's the most common cancer, yet the mortality rate is not as high as lung cancer? Again, the credit goes back to the NHS. We have a screening program that every single woman after a particular age that they go through, again, being NHS, it's free of charge. So it's the early detection. That's what helps. And we know that it's directly related to the survival. So that's about breast cancer. Now, bowel cancer, again, it is the fourth common cancer in the UK. The number per year, new cases, it's 42,900, not great, still high. And if you look at the mortality, it is 16,800. If you compare it with lung cancer, it's not as high as lung going on, which really helps with early detection. Finally, we have prostate cancer. This is again about 52,300 every year, and it is the most common cancer in men. We see that a lot. We know that men don't like to talk about things like this. They leave it too late, but the awareness is there. Men are starting to come talk to their GPs. There are quite a lot of programs out there to increase the awareness in men. So all that is working. So there are around 12,500 prostate cancer deaths in the UK every year. Thank you. Next slide, please, Kirti. So let's come to the cancers that are linked to genetics, your forte. We've got a long list, but if you look at that, especially we spoke about those four common cancers, and it's quite interesting to see how a few of them are linked to genetics, and that's why I thought it'll be more fitting to talk to you guys about it. So breast and ovarian cancer, colonic cancer, Cowden syndrome, gastric cancer, Gorlin syndrome, men, neurofibromatosis, Peutz-Jeghers syndrome, pheochromocytoma, retinoblastoma, von Hippel syndrome, and Wilms tumor. Next. 10% of the breast cancer cases are thought to be hereditary, and the most common cause of hereditary breast cancer is in an inherited mutation. You guys might know about this, the infamous BRCA1 and BRCA2 genes. On average, a woman with a BRCA1 or BRCA2 gene mutation has up to a 7 in 10 chance of getting breast cancer by the age of 80. So you can imagine that is 70%. 7 in 10 of them actually have a chance of getting breast cancer if they do have this gene. Moving on to ovarian cancer, again, a variant in the BRCA1 and BRCA2 genes account for most hereditary ovarian cancers. About 15% of women, we are talking about 15 in every 100, who develop ovarian cancer have a variant in either their BRCA1 or BRCA2. And there are variants in the other, like the RAD51C, 51D, and BRIPI genes can increase the risk of developing ovarian cancer. But these variants are rare. A little bit about the bowel cancer. This is something we see a lot. My daughter, she again is a general practitioner, and we were talking about this cancer and genetics the other day, and she mentioned that in her list, they have quite a few patients with Lynch syndrome. And that is really, well, worrying in the sense that when you look at the prevalence, but it is good that it's been actually diagnosed and patients are aware of the diagnosis and what can be done about it. Okay, so the Lynch syndrome itself, a person who inherits Lynch syndrome will have a high lifetime risk of colorectal cancer, which we do see a lot, as well as an increased risk of some other types of cancer. It can be caused by variants in 1 in 4 different genes, like the MLH1, H2, MSH2, H6, and PMS2. The code for essential DNA repair protein. In all 4 genes, only 1 affected copy is needed. As you all know very much, this is a dominant pattern in families. You also have the MAP, another inherited syndrome that increases a person's risk of developing colorectal cancer, and the MUTYH associated polyposis, which is more often autosomal recessive. Now, this next one, I have actually seen patients or babies with retinoblastoma, and it really gets to you. It does when you see a baby, because we do newborn screening tests at 8 weeks. So part of that screening is to have a look at the eyes, and I have diagnosed, well, not diagnosed, seen patients referred and later who went ahead to have a diagnosis of retinoblastoma. So retinoblastoma, it is a malignant retinal tumor that affects young children. Mutation in the RB1 gene causes retinoblastoma. Almost all cases of retinoblastoma occur due to variants in the RB1. Next slide, please, Kirti. So we spoke about all the statistics and the numbers and you know how it actually clinicians actually see patients on a daily basis. I see about 30 to 35 patients a day, and I am guaranteed to see at least 1 patient, minimum 1 patient with cancer. So when we are talking about that high numbers, so what is this NHS doing about it? Okay. So we have well, the NHS has come up with quite a few cancer pathways. So what is a cancer pathway? It is the patient's journey from the initial suspicion of cancer, starting from when you see the GP with the suspicion of cancer, through the clinical investigation, and then the patient diagnosis and treatment. So what happens when the patient comes in and sees the doctor, and the doctor suspects that probably this is cancer? Then immediately the patient is put on something called a 2-week wait pathway. What it means is that right from the time the decision is made to refer a patient, to investigate for cancer till the patient gets the test. That should be done within 14 days. And that's the 2-week wait. So the 2-week wait appointment system was introduced so that anyone with symptoms that might indicate cancer could be seen by somebody who's an expert as quickly as possible, which is 14 days. The target for all cancer treatment pathways is for at least 85% of the patients to start their first treatment for cancer within 2 months of an urgent referral. 62 days might feel a bit longer, but we must remember that we are talking about a big organization, free of cost, and all those things. So what happens? So you've got your patient, you've diagnosed the patient with cancer, what next? So there is a big multidisciplinary team that works around this patient and makes sure that the patient gets the proper right care. So people who are involved in multidisciplinary team are the GPs, specialists, that includes oncologists. Then once the patient is back in the community, you have the community nurses who are called the district nurses. You have the Macmillan who are the cancer specialist nurses. You have the occupational therapist who goes to the patient's home to make sure that they have everything. For example, you have your colorectal cancer patients, then you need to make sure that they have the commode and things like that. Are they in a bungalow? Are they in a house? Can they walk upstairs? Is there a stair lift? Everything that's being done by the occupational therapist. Then you have your physiotherapist and the social worker who makes sure that they have a good care package from, do they have their finances sorted, who's going to pay for their meal, how are they going to, so they take care of pretty much everything. So this whole team works around the patient after the diagnosis of cancer. So then not every story has a happy ending. So when there are patients who go into palliative care, so the palliative care pathway kicks in. So what is palliative care? It's a specialized medical care for people living with serious illness, such as cancer, but not exclusively cancer. It can be for patients who are in their end of life due to heart failure and those sort of things. Then finally, you have, well, when it comes to the end, it's just end of life pathway, where all you're doing is just making sure you stop the no active intervention, but keeping the patient just comfortable by giving them the anticipatory medication. And all this is done by that multidisciplinary team. So that's the cancer pathway. So what are the recent developments? This is just an ongoing thing, and this is not something new. We've always had cancer in the community, but all we know is it's just getting bigger. So genetics, as you and I know, usually refers to the study of the single gene disease. However, genomics, on the other hand, it refers to the study of the entirety of the person's DNA, which including the genes and the DNA in between, which is the genome. So what did NHS do about this? So NHS came up with this genomic medicine service, which was launched in 2018, not too much in the past. Those of you who are really interested in this, I would direct you to this website, and there's plenty of material that you can read. It's quite interesting. This is just going to be in my PDP for my next appraisal. I was quite fascinated. So what did they do? The transformational 100,000 genomic project sequenced about 100,000 genomes, adding significantly to knowledge of cancer and rare disease. So this is what is really interesting, the rare disease, which we will talk about in a minute, and providing more patients with diagnosis. So this was the first step towards establishing a genomic medicine service. NHS had not done this before. And increasing the use of genomic information within routine. We're talking about routine NHS patient care. When the patient just walks into a GP surgery and they can talk about, ask questions, and the clinician is expected to give them some information, or at least to refer them to the appropriate people. And that's known as mainstreaming. So what was the aim of this project? The main aims of this project were to make genomics part of routine healthcare. Watch this space, guys. You will be surprised. In the years to come, we are talking about another 20, not even 20, I'm thinking of 10 years, like people walking into a GP surgery, walking into a doctor's consulting room, asking for, I would like an ECG, I would like a blood test to get my blood sugar, blood cholesterol tested. People are going to walk in asking for this genetic testing. That is the future, and that is you. And the other aim of the project is enhanced genomic healthcare research. That's where you come. Plenty of opportunity for the ones who are in this field. If this is your specialty, a lot of opportunities out there. And also uncover answers for participants. There are plenty of patients with symptoms and simply with no diagnosis, and just clinicians are doing symptom control. So we need to provide answer for these patients, and these were the aims of this particular project. Next slide, please, Katie. So what they do, they looked at the genes of about 4,660 people from 2,183 families. That's a lot of families. But what did they do is, with rare disease who were early participants in this genome project, and they were analyzed using this is the future, the whole genome sequencing. And they've done some brilliant work on that. The results showed that using this whole genome sequencing, that led to a new diagnosis of 25% of patients. So what we are talking about is 1 in 4 patients were out there with symptoms, suffering without a diagnosis. What that means is, if there is no diagnosis, we can't treat them. So we were actually letting people down, letting general public down, because we didn't have the knowledge. So that is where research is so very important. And what it did to this 25% of patients is amazing. So many of these patients had gone through years and years of appointments. The number of things that they must have undergone, the number of drugs that they must have been given just to as a trial, the number of side effects, just because they didn't have a diagnosis. And what a good boon it must be for these people. Now, again, this WGS enabled them to not only get a diagnosis, pave the way for treatment, and save on vital health resources. Remember, it's all free of charge. That's why the emphasis is so much on evidence-based, cost-effective treatment in NHS. So, for example, there was a 10-year-old girl whose previous 7 years search for a diagnosis had multiple intensive care admissions, over 306 hospital visits at a cost of £356,571. Just imagine, leave all the money out of it and just looking at a 10-year-old, a normal 10-year-old, what do we expect? Go to school, come home, have a good play, go to bed. That's normality for a 10-year-old. But here you had a 10-year-old girl going through 7 years. So we are talking about pretty much from when she was born, having intensive care admissions, 307 hospital visits. That's saying something, isn't it? So genomic diagnosis enabled her to receive a curative bone marrow transplant at a cost of just £70,000. In addition, predictive testing for her siblings showed no further family members were at risk. And what a relief that must have been for the parents. So the pilot study showed that this whole WGS can effectively secure a diagnosis for patients, save the NHS vital resources, and pave way for other interventions. Next slide, please, Kirti. You really need to. Because next will be a real life story. I'd ask you to take a couple of minutes to actually listen to what these two families went through. Thank you.
So it was a genome sequencing test to determine what type of cancer it was and if the cancer was genetic. And we found out that it wasn't genetic, which was a massive, massive relief.
That test was the key to everything. That solved everything. Oh, without that testing, it would have been a completely different story.
Yeah,
I mean,
we would have gone through chemo.
So we did the biopsy, we sent off material for the normal testing, the normal standard of care, and then we also at the same time sent off stuff for whole genome sequencing, some tissue, which now in England is also standard of care. And at Addenbrooke's, we offer to every child who walks through the door.
It was an emergency diagnosis, really, that she had the cancer, even though we didn't know what type. But obviously our consultant knew after doing the tests, and we've been blue lighted to Addenbrooke's.
Well, you think it's a nightmare, really. You don't think it's true. You just think you can't believe it's happening.
We
really,
look
more
back
than
and
anything.
it's just a big blur, really.
Yeah. So Aubrey presented to us right at the beginning when we were starting doing whole genome sequencing and offering it as standard of care. So we sent her tumour off. Now, under the microscope, it was very difficult for the pathologist to tell us exactly what her tumour type was, and there were a number of possibilities. And that made treatment at the very start of her journey challenging to work out what was best. But quickly, with the whole genome sequencing, we got the results back. And what that showed was that her tumour was a specific type of sarcoma called rhabdomyosarcoma. And by looking at all of the information in her tumour, we were really able to tailor her treatment going forward once we got that information.
In Oliver's tumour, we found a mutation in a gene that's called PDGFR beta. It doesn't really matter what it's called so much, but once you've got a mutation in that gene, you completely leave the territory of cancer, and then what you think about is a benign tumour called a myfibroma. What was so astonishing about the results was this. The type of mutation was a very peculiar type of mutation that we could not have picked up with any other assay.
To have that testing, we didn't even need to discuss it, did we, really? It was
offered to us, and we literally instantly just said, yes, go for it.
It meant that me and Paul sort of knew we hadn't passed anything down to Aubrey, and it also meant that we know our son Harry, who's just about to turn 7, is not at risk of getting this cancer also.
The whole genome sequencing information, along with the pattern of where her disease was on scans, told us that chemotherapy was the best option for her and avoided her going through.
Thank you. It's just a little bit more about what they've been doing. So I'm just quite mindful of the time. We are coming to nearly the last 5 minutes here. So I'm just quickly going to go into the future of genomics. So what can we expect and what are the opportunities out there? If you move on to the next slide and the one after that, please. So what have they done in the UK? Come up with this Genome UK 25 implementation plan. So in October 2022, just 3-4 months back, NHS England published the first ever genomic strategy, which is accelerating genomic medicine in the NHS, which sets the strategic direction and priorities over the next 5 years. The aim of this is to ensure that genomics will be at the heart of the sustainable NHS in the future and the next generation of healthcare in the NHS. So it's all about the next generation. So there are about 44 objectives. I'm not going to bore you with all those, but a few of them being mainly to provide rare disease and cancer patients with an accurate diagnosis earlier and more quickly, whilst also supporting the use and development of a personalized treatment. This will also include a Cancer 2 program, which will be using this artificial intelligence to analyze the genomic data alongside the digital histopathology and radiologic images, so that you can give an accurate diagnosis and speed the diagnosis as well. And also extending the use of the treatment, the pharmacogenomics in the NHS will ensure more patients get the right treatment at the right time and the right dose, so there are no side effects, there are no adverse events, improving the outcomes for the patients and reducing the number of these. Using genomics to better understand the pathogens and how they spread. We've all been through the pandemic, so we know what a devastating effect it can have, as well as the role of the person's genome in the response to the infectious disease. This supports scientists in controlling outbreaks. Enable the NHS to move forward from a system that primarily detects and treats illness to the one that utilizes genomics to predict and prevent ill health. So, coming to the last slide, next slide please. So we started off by saying how it is all gloom and doom, this whole cancer, but I'd like to end with a happy note, how the whole research, what it can do. If you could really play this clip, and hopefully we'll get the audio this time. Thank you so much, Keithy.
Thank you so much for your time. And if you're having some questions, maybe I'll message you and you'll be able to answer it.
Absolutely. And guys, if you have any questions, please do let Keithy know, and I'll be more than happy to answer your queries. Thank you. Thank you very much. Bye. Bye, everyone.
Thank you, sir. Thank you so much.
Okay, thank you. Okay, good afternoon to all. I am so happy to introduce one of our chief guests, Dr. Satish Kumar Rajasekaran sir. He is working as senior postdoc researcher in Rishon LeZion Institute in Israel. He completed his UG degree in biotechnology from SRM University in the year 2006. He completed his MSc degree from Madras University in the year 2008. In his PG degree, he got university third rank, and he completed his PG diploma in molecular diagnostics from Allahabad University in the year 2009. In his PG diploma degree, he got university first rank, and he completed his PhD in biotechnology from Christian University, Tanjavur in the year 2016. He received 2 patents from Korea and he published 34 research papers in the reputed journals, and he is also first author in 23 reputed journals, and he also called invited speaker in the 4 national conference, and he gave 4 oral presentation and 2 poster presentation in the national conference, and he received excellent postdoctoral fellowship of ARO from Israel in the year 2020, and he also got JRF fellowship from Government of India in the year 2015, and he has academic collaboration with Chonam National University and Budik University and Yeongnam University from Korea, and thank you to all.
Okay. Yeah. Thank you for the introduction, and as it is already delayed, I think I will straight away proceed with slides. So I don't see the, yeah. So shall I begin? Yeah, okay, fine. That's good. So before I begin, I would like to say that the topic which I'm going to present right now is quite not very close to the conference overall theme, but I'm sure that I'll try to keep you intrigued, and most of the topics are informational, so it might benefit everyone who is listening. So the topic here is what you see on the screen is probiotic mystery with the volcanic community of lactobacillus cells. So indeed, the topic is ambiguous, and it's a bit of it will be confusing because it. I will try to clarify each and every points when we go through our slides. So before we go into the research slides or some of the figures or the publications which we have authored, I would run through a few terminologies to make the picture more clear. So the first slide here, what you have is probiotics. What are probiotics? For instance, probiotics are live bacteria with a non-pathogenic and can be beneficial to the to humans. So nowadays most people or most patients consume probiotics as supplements. So what you see is here is probiotic is a live bacteria and is beneficial. And when these probiotics form biofilms, I'll explain what are biofilms in the next slides. They are called beneficial biofilms. Yes. So here you will know what exactly is biofilms. Biofilm is something which it's a sessile community formed by bacteria that attaches to the biotic and abiotic surfaces. So in simple terms, if the biofilm are harmful if they are caused by bad bacteria, and they are good if they are caused by good bacteria. So for human health. So if you see the figure here, it's an example of a dental biofilms. Biofilms are, in short, they are sticky substances where bacteria, a mesh of bacteria which are shielded from antibiotics and the host immune system. So the bacteria inside the biofilms are quite resistant to any immune cells or antibiotics. That way, it's very hard to remove the biofilms. For instance, the dental plaques. If you this is a biofilms formed on the tooth. So this is an example of biofilm formed by Streptococcus mutans. So in short, to be simple, if a good bacteria forms biofilms, it is beneficial, and that is a good bacteria means the probiotics, and it can be harmful. The same biofilms can be harmful if it is formed by the bad bacteria or pathogens. Next slide. Yeah. So the concept here, which we are working on, is to use positive biofilms or the biofilms formed by the good bacteria to mitigate the biofilms formed by the pathogen bacteria. So there is a competitive exclusion principle going on here. What happens is that if there is a bad bacteria in our system or in our body, we try to take probiotic supplements. When we take probiotic supplements, what happens is that the probiotic bacteria or the good bacteria which is in the supplements go and compete with the pathogenic bacteria, which has already creating problems in our people are known as nomads. So similarly, Lactobacillus plantarum has the trait that it is widespread throughout the ecology. You can find Lactobacillus in plants, in mice, in soil, everywhere in the ecosystem. So that's why it gets the name nomadic. So among all the Lactobacillus species, only 3 of the Lactobacillus species are classified under nomadic. So there are 3 different lifestyles. You have free living, nomadic, and host adapted. But the nomadic one is, it has a lot of adaptational advantages. So that is the reason why Lactobacillus is specifically considered a very good source of probiotic, because it is available throughout the ecosystem. Yeah, next slide. So yeah. So straight away now I'm showing you some of our very important findings. You see all the findings in a nutshell. So we used Lactobacillus plantarum that was isolated from the cows. And in the laboratory system, what we see is that we saw 3 different unique novel phenomena which was not revealed or identified before. For instance, I have marked the phenotype 1, the isolates from the Lactobacillus, isolates from the cow, where exactly they were able to form cone-shaped morphology, cone-shaped colonies on hard agars. You can see the cone shapes. And then during cold response, they formed a lot of aggregations and a lot of cold shock bundles. What we call is that biofilm bundles. And finally, the phenotype 3. I'll explain about each and every phenotype and I'll show you the results. So finally, when the last phenotype is that when you expose this to low pH stresses, what happens is that it entirely changed the geometric structuring. You see, usually pH of pH 5.5. And what we understood is that everything is regulated by pH, is controlled by pH. When you increase the pH to pH 7, that is neutral pH, you don't see the cone shaped, although you see the cone shaped, but there is a tip abolishment, and there you see there is a change in the pattern. There is no coloration, there is no brown coloration, and there is this justifies that at acidic pH they are adapted to external stresses. They have brown coloration and they are not vulnerable to any other further stresses. So this was one of the phenotypes. Although I'm not showing all the results, I'm just showing the major images. Next slide. So what we do here is exactly we use that pH 5.5 grown cone-shaped colony cells, and we stress them to by placing them in minus 17 degrees Celsius. What happens here is that it's a cold stress. When you apply a cold stress to the cone-shaped colonies, what we saw a new phenomena which we termed as the sacrifice for survival phenomena. So what happens here is that a lot of during a cold stress exposure, a lot of bacteria come together to form biofilm bundles. What you see on that, and what we termed is called consolidated bundles. You see red and green bundles. The green is actually the live cells, live bacteria, and the red are the dying or the dead bacteria. So cold stress actually kills the bacteria, but somehow a certain group of subsets survive. And how they survive is because the dead cells or the dying cells are shielding them within the bundle, and then that's how they recover eventually after the stress. So it's a sacrifice for survival phenomena, first time reported by our group. So yeah, that's it. So we further try to understand the mechanism of sacrifice for survival mechanism, how it is regulated. And when during this we try to understand what could be the genomic pathway that might regulate this sacrifice for survival system. So what we learned is that Lactobacillus plantarum, as I said earlier, it is nomadic, and they have a specific set of heat shock protein genes, which is not found in other lactobacillus. So that makes Lactobacillus plantarum unique. They have a set of 3 sets of heat shock proteins gene, HSP1, HSP2, HSP3. So these heat shock HSP genes are not found in other lactobacillus species. So we used the mutants of heat shock proteins HSP1, 2, and 3, and found that HSP1 that is linked to cryotolerance, when this gene is mutated, we don't get the cold shock sacrifice for survival phenomena. If you see the bundles in the wild type, WT is the wild type. If you see the wild type, you have the mixed bundles where live and dead cells are there, red and greens are shown. But in case of HSP mutants, what happens is that most of the cells die. That makes the gene very important for the sacrifice for survival rescue phenomena. So we identified the gene that is responsible and is the candidate for cold shock stress response via this. Thank you. Next slide. Next slide. So there were these are some additional these are some additional parameters. If you see that so you see what happens is that it's not just the during cold shock response. What happens that certain signals also activate, and these signals or signaling molecules are present in the cell-free supernatant. And then this cell-free supernatant when we tested the cell-free supernatant, that these signals can induce these cold shock bundles in other bacteria as well. For instance, we work with Bacillus subclassis model also, because that's one of the common model we use for generating mutants. So in this case, when we tried with Bacillus subclassis, what happens is the signals from Lactobacillus plantarum was used to enhance the biofilms of Bacillus subclassis. So there is a co-species interaction taking place. There's a crosstalk between Lactobacillus plantarum and Bacillus subclassis. That makes what happens is that so far we were using only one kind of probiotic, using only Lactobacillus plantarum, but now we recommend the use of a combination of Bacillus subtilis, because Bacillus subtilis is also a potential probiotic, and when in combination, it is easy to fight the bacteria, the bad biofilms, which I talked about earlier. Yeah, next. So right now we are trying to illustrate the pathway and trying to find what exactly are the signals. Next slide. Finally, the most intriguing phenotype is still it's we have the phenotypes. So was the formation of V-shaped conformation. If you see the difference in pH 5.5 and pH 3.5, you see the cells. There are only single cells in case of pH 5.5, but in case of pH 3.5, what happens is a cell takes a new form. cells takes a new form, because if you note the probiotics was first coined in 1919 by Erich Mechnikov, as a Russian scientist, and he was the first one to identify probiotics in yogurt. And this was done with there was a bacteria called bifidobacterium, which also had a similar Y-shaped structures. So there is something with the structuring of the probiotic, which is really important for its activity later. So here we demonstrate for the first time a V-shaped conformation, and what we see is that V-shaped conformation structure is a set of 4 undivided cells. But why that's happening is that's what we try to elucidate further, and we found that, next slide. We found that these V-shaped cells triggered positive biofilms. If you see the biofilms, on the lower panel, you will see the biofilms on polystyrene surfaces. What happens is that at pH 3.5, you have a thick biofilm stained with cyto9. That's why it's green, a fluorescent green. In pH 5.57, you don't see a lot of green. That means there is less bacteria, so there is less biofilms. So what you see here is at pH 3.5, when the cells form the V-shaped structure, it actually triggers biofilms. So this is a new phenomena of generating positive biofilms via V-shaped conformation. Next slide. So next we try to find the significances of the V-shapes. What happens is that the V-shape, we try to do a competitive assay with a pathogen. We used Candida albicans. It's a human pathogen you have in dental biofilms and several other. It causes, it's a serious yeast infection. So we actually co-cultured. Try to study the interaction between Lactobacillus plantarum and the V-shaped structures and Candida albicans. So when we understood that the V-shaped structures were significantly eliminated the biofilms from the Candida albicans. You see, in the control of Candida albicans, you have a dense green biomass, but in case of Lactobacillus treated with 3.5, you don't see Candida albicans biofilm. So we have proved our concept that positive biofilms, a newly generated via the V-shaped geometric structuring, is able to remove the biofilms of Candida albicans and can be used as effective probiotics in the near future. Next slide. So next was to we did them. We just scaled up to the model and we used C. elegans. C. elegans is a nematode. It's a nematode which can be used for toxicity testing and other research parameters. This is economical because instead of going to the mice model, you can work with the C. elegans. So one interesting approach is that Candida albicans, when you treat Candida albicans or feed, exactly that's the term to be used here. If you feed Candida albicans to the C. elegans, the nematodes, the pathogen will kill the nematodes within time frame. But at the same time, if you co-culture with the V-shaped structure, that's what we did here. What happens is that it is able to prolong the lifespan of C. elegans. Lactobacillus, the V-shaped cells are able to prolong the life cells of Candida albicans infected nematodes. So this is a proof of concept that that our V-shaped structures are working fine in a living model. Now next we can always recommend for the human model. Yeah. Next slide. So straight away we are going to the conclusion. Actually, we in this talk I have briefed about some of our various three different phenotypes which we identified, and one of them, the V-shaped structures, can be used for, has a lot of applications. For instance, biofilm inspired encapsulation technology to bring up symbiotic food. Symbiotic is a combination of probiotic and prebiotic structuring. So there are a lot of applications of V-shaped structures. You can even lyophilize it and provide as a normal supplement. You can combine with Bacillus subtilis, use as a co-supplement with 2 different probiotics, and they're an option. So right now we are working on a similar pattern with Bacillus subtilis. You can see the cone-shaped structures that Bacillus subtilis erases or forms on agar plates. This is actually sort of a novel finding, and we are now trying to knock out the mechanism which is closely linked to the Luxus mechanism. Luxus is a quorum sensing mechanism. Okay, thank you very much. And with this, I should end the slide. And I thank all the organizers. I thank Dr. Nidhi Rajan, sir, and Kirtana. Thank you for your.
Good afternoon, dear students. Yes, so we have come back to the last session of the whole 2 days international conference. So this will be the last technical session, and after this we'll be having a valedictory function. So for the last technical session, we are having Dr. Subbu sir with us.
Yeah.
Okay,
Sorry.
and to chair the session, we are having Dr. Shobana with us. And about Dr. Subbu sir, he's a scientist from Sericulture Institute, and he'll be sharing his knowledge with us. And with this, I'm handing over the mic to Dr. Subbu sir. Kindly please take over the session, sir.
Myself, Subramaniam. I'm scientist at Seri Biotech Research Lab. Topic of the presentation for this particular topic is molecular characterization of novel RNA viruses infecting wild silkworms. Actually, the main objective of this particular presentation is I wanted to make aware of what are the important molecular tools. By using that, we can address a disease problem. The other thing is how to do a research systematically to address a scientific problem, which is also a rationale in this your workshop. Madam. Yeah. So first, I would like to show overview of sericulture in our nation. So you all know that sericulture is nothing but cultivation of silkworms for silk production. Okay. So my presentation would be a kind of hybrid. We have few basic slides followed by little deep into the subject. So in this particular slide, I wanted to tell you that in our nation there are 5 different kinds of silkworms. The first one is Bombyx mori. The second one is Samia ricini. We call it as Eri. The third one is Antheria assamensis. Antheria milata, the fourth one. And the fifth one is Antheria praili. So among all these things, these 2 Bombyx mori, that is mulberry silkworm, and Eri silkworm, these 2 are domesticated ones. What do you mean by domesticated? We can rear or we can cultivate these silkworms in our homes. However, the other 3 ones, the 3 insects we could not rear in our home. These are wild silkworms. We could able to rear only on trees. So especially in the northeastern regions, we have their food plants. These are insects, obviously they eat food. That food comes from trees. These 3 particular silkworms will rear only on trees. And again, as you could see, these are the cocoons of respective silkworms, and this is the silk yarn. And here I have shown the, as for 21-22 recently, in our country, we have 25,000 metric tons of Bombyx mori. So the cost is 1 kg how much? This is 4,900. So it's like this. Second one is Eri. So the cost is around 3,200 for kg. So we have around 7,000 metric tons. 1 metric ton is equal to 1,000 kg. Okay. This is just a overview of Indian sericulture. You just look into this figure. Here 1 kg is 32,000. It's kind of a gold. We call it as a golden silkworm. In the entire world, only India has this particular type of silkworm. We can find in Assam, Meghalaya, Tripura and Nagaland regions. Nowhere we can find. We are proud to be, this is the pride to be in India. We can find these silkworms only in India, and this silk is also like a lustering or glittering golden color. The cost is around 32,000 just kg. If you can able to produce 2 kg per a month, the farmer used to get 72,000 rupees. So that government of India is giving a lot of impetus, and they are giving different kinds of societal schemes for enhancement of this particular silk. So the next one is Mylita, 4,700 is the kg. This is 1466. So here you could see, though this is very costly silk in the world, but the production is very less. Why the production is very less? Because these silkworms are confined into a particular geographical zone which is located in the northeastern region, okay, mainly in Assam and Meghalaya and Nagaland. The last one, this is Voktasa silkworm. The price is 9,000 kg. So I must tell you that, so there are 8.8 million people are employed because of the sericulture. It will give you a lot of employment, okay. And how big is this industry? In India, we have 1.7 lakh crores is correspond this industry. So 1.7 lakh crores per year is involving for this sericulture activity. Therefore, Government of India has started Central Silk Board. I am from Central Silk Board. The Central Silk Board actually looking after production and productivity activity of all this silk among the nations. Like a DST or Department of Biotechnology, we have Central Silk Board. All over India, we have 20 different kinds of research institutes. I am coming from one institute that is located in the Bangalore. Okay, so with this brief introduction, I would like to go further into my slides. See, madam, go back, please go back. Yeah, since these particular 3 silkworms are being reared outdoor, they are exposed to different kinds of pathogens, right? If they are exposed to different kinds of pathogens, obviously the farmers lost the crop. So we would see different kinds of diseases, how you characterize a disease. So what are the molecular tools, and all we will see in other slides. Go ahead. So in my presentation, I am going to speak about how to characterize viral pathogen which is affecting silkworms. As you could see here, so virus is nothing but a nucleocapsid. So please pay attention on this. You may have in your subject virology. I just give you few fundamentals. Later we will go deep into subject. The first one over here, this is just a nucleic acid. Virus is nothing but a nucleic acid, and it is actually covered by a proteinaceous layer. This proteinaceous layer, what you see round color circles, is known as capsomers. All these capsomers form a capsid. This entire structure is known as capsid. Individual, this circle is known as capsomers. Okay. So this capsid plus nucleic acid is nothing but a virus. That is a nucleocapsid. Okay. So this is also virus. What is the difference between this and here? Can anyone tell? The difference between these two structures. Yes. Here what happened? This is a nucleocapsid, nucleic acid surrounded by a proteinaceous capsid. Along with this thing, there is one more a protective layer. This is known as envelope. Generally, this particular virus gets this envelope from host. Okay. So in the nature, we have only 2 different kinds of viruses. One is naked and the other one is envelope. Naked means just a nucleocapsid. One nucleic acid surrounded by a proteinaceous layer. Whereas here, nucleic acid surrounded by a proteinaceous layer, in turn again protected by one more a lipid bilayer known as envelope. So in my later slides, I used to speak about naked. Then you have to remember that it is just like this structure. Okay. Go ahead. So this is little deep into that. See, in the earlier we have a nucleic acid and a capsid. In turn we have a small proteinaceous molecules. We are calling it as virus attachment proteins. So these are very much crucial for the virus to attach host cells. Okay. So these are located at the capsid, right? Here also we can see virus attachment proteins. Along with this thing, we have a small matrix proteins. So whatever the proteins which are associated in the virus structure is known as structural proteins. So whatever you are watching here, all are nothing but structural proteins. So the structural proteins are very much important to characterize the virus, right? So for example, here what are the structural proteins? Capsid is a structural protein, and again we have embedded proteins known as virus attachment proteins. Those are nothing but a structural protein, and again here we could see matrix proteins. These all are structural proteins. If you want to identify or characterize a virus, first you should know what are structural proteins and what are non-structural proteins. I will come later what are non-structural proteins. Please go ahead. The next, here see, in insects, a particular type of virus forms a proteinaceous matrix known as polyhedra. Polyhedral bodies we could say. This is single virus. All these virus to produce a matrix protein, this is known as polyhedral body, as you could see here. So this polyhedral body will give you a protective cover to the virus. What happens basically, if the virus is exposed to sunlight as UV rays, right? These UV rays are very much dangerous to virus capsid layer. But if the virus is in this particular polyhedral body, they can withstand temperature, they can withstand pH and humidity also. Any polyhedral forming virus can withstand up to 2 years in the natural environment. So this type of virus we can generally find in sericulture. If any virus is forming such type of structure, we are calling it as accalsion body or polyhedral body, as you could see here. Now symmetry. Symmetry means what happened? In the second slide, in the structure of the virus we have seen, virus is nothing but a nucleic acid. So nucleic acid surrounded by a capsid layer. How this capsid layer is arranged? Based on the arrangement of capsid, we have 3 different kinds of viruses. The first one is helical. This is rod shape. This is nucleic acid surrounded by protein, capsid protein. In the form of helical fashion, we are calling it as helical. But sometimes you can see such type of structures, geometrical structure, we are calling as icosahedral. Okay. And the third one is complex. So there are only 3 different shapes of viruses we can see in the nature. One is helical or maybe icosahedral. The third one is complex set of virus. So almost all the viruses which affect silkworms comes under this category, icosahedral as well as helical in nature. Now, if you want to start any research, it is very much important to collect the literature. What is there in the literature already? So we have to look for the literature, compile the data so that you will understand what is the research so far happened, what are the gaps and what we have to do. So as for I already told you that most of these wild silkworms are susceptible to virus diseases. So obviously if I want to do research, I wanted to know what type of work so far happened. So we have collected literature. Based on the literature, we have classified the pathogenic, insect pathogenic viruses. As you could see here, occulted virus, non-occulted virus. So what is meant by occulted? As I already shown, if the virus is forming a matrix protein and inside it will reside, that is known as occult virus. Non-occulted means just a nucleocapsid. And again, I told you that like humans, plants, animals, viruses also has its own genetic material. What is our genetic material nature? What is our genetic material? DNA, right? Virus is also having DNA. There are some RNA viruses, again in turn single stranded RNA, double stranded RNA. So based on the genetic content as well as the structure, we have 4 different kinds of viruses over here. So one is double stranded DNA. This is a baculoviridae, we call it as. The second one, double stranded RNA, we call it as reoviridae. And then we have single stranded DNA and single stranded RNA viruses. As you could see here, this it's a yellow color, cypovirus and inflavirus. This particular information actually from our lab we have generated this. This is the overall picture of different viruses we can find in the sericulture. Yeah, this particular table will tell you different kinds of viruses, the disease and the corresponding crop loss. Now, I will tell you, as I already tell, these antherius species are known as wild silkworms. They are susceptible to different kinds of viruses, diseases. As you could see here, their symptoms. This is domesticated one. As you could see, there are 2 different viruses. One is a bidensovirus and a nuclear polyhedrovirus. Whereas here in Assamensis, we have cypovirus. In Myleta, we have inflavirus, and priorly we have again NPV virus. Now I will tell you, these are the characteristic symptom of virosis in Muga silkworm. So earlier, we used to get symptoms like this. We were not knowing which virus is causing this particular disease. And this is the one of the costliest silk in the world. Okay, 80% crop loss. That means that once this disease comes in the rearing field, you will lose 80% of your silkworms. Only 20% silk will survive. So we know this is a disease, but we don't know the causative agent. So now the research starts from here. We want to identify which is the virus which is responsible for this particular disease. So what happened? As you could see here, first this is the control one. You can see progut. This is the gut, mid-gut part of the insect, and you could see the viruses affected one. You can see kind of ulcers, and also entire mid-gut is actually calcified. They looks like white color. Okay, this is a hallmark of the virosis symptom. So as you could see, these all are dead worms. These all are dead worms because of the viruses. They are falling down. Okay, these are the healthy worms. Would you be able to see the figures? Can anyone see the figures? Clear? Okay, now why we want to identify which virus is causing this type of mortality? Because as I already tell, one single silkworm cost is around 10 rupees now, right? So you could see how much crop loss is a farmer is facing over here. So the first step is, we know this is a virus. So first and foremost important step is we have to go for microscopic analysis. The first one is light microscopy. So in the light microscopy we can see polyhedral bodies. These are polyhedral bodies, this round color, right? This is a scanning electron microscope followed by transmission electron microscopic studies. So viruses are very ultracellular organisms we could not see by our naked eye, right? So we have to through TEM through only we can able to see. This is transmission electron microscope. This is the midgut portion of the insect where you could see different kinds of polyhedral bodies. So this is at 5 microns resolution. If you can go further, if you can resolve 0.5 microns, this is the polyhedral body. In between we could see virions, individual virions. Here it is very clear. You can see these are the dots. Each dot corresponds to a particular virus, single virus. These all are virus. Don't make noise, please. Okay. Yeah, you please go ahead. Then, if you could compare this structural comparison, we have two different viruses. One is NPV. So earlier we have reported this virus. You can see these are the rod type structures. But here, which is we are finding in the Moga silkworms, we don't have this type of rod shaped structures. Instead, it has polyhedral bodies, including have a small dots. This dots indicates a spherical icosahedral virus. So in the classification of the virus, we have seen already. Go ahead. See, they are forming polyhedra. That means that they are occulted virus. So no need to do any studies regarding non-occulted virus. We have to focus only this particular portion. Now in the occulted virus, the TEM, transmission electron microscope, clearly indicated that this is not baculoviridae because baculovirus forms rods, but it is showing small dots. So which virus form a dot-like structure in the transmission electron microscope is nothing but reovirus. But this is just a structural morphology. We want to prove molecular-biologically, yes, this is a reoviridae or cypoviridae. Go ahead. Then what happened? We have extracted, first we have purified the virus, extracted genetic material. As I already told, like humans, virus is also having genetic material. The genetic material may be a DNA or maybe a RNA. So once you extract genetic material, immediately we will come to know whether it is a DNA virus or RNA virus. As you could see here, we were unable to extract DNA from the purified viral particles because this is not at all a DNA virus. We used to get only RNA. The RNA looks like this. It's a segmented RNA, not a single molecule. There are 11 different segments will be there. So this is a segmented RNA occulted virus. Like this now slowly we used to gather the information regarding this particular pathogen. As you could see here, this is the RNA. It's not a single molecule, segmented RNA. Later slides I will show you. Then what happens, since we know the information, this is RNA virus, then we have designed different kinds of primers to amplify structural as well as non-structural genes. What are structural genes? Structural genes actually encodes structural proteins, right? So whatever bands we could see, these all are structural proteins. One is non-structural protein, that is RDRP, RNA dependent RNA polymerase. Like humans, we have DNA. Our DNA is replicated by DNA polymerase. Same thing, virus is having what? RNA. RNA has to be replicated by an enzyme that is known as RNA dependent RNA polymerase. So then what happened? We know these bands are there, but we don't know what is the sequence of these bands. We have to clone these bands, then send for sequencing. Based on the sequencing information, we will come to know which type of structural protein is this, which type of RDRP. So all this homework has done, and we have to submit all this sequence into database. Then it is found that this is nothing but a SIPO virus. It is occluded virus, double-stranded segmented DNA is the genetic information. It is having RDRP, that is RNA dependent RNA polymerase. Please go ahead. Now see, there are 22 different kinds of SIPO virus. So far we came to know this viral pathogen is a SIPO virus. It is fine, but which type of SIPO virus is this? There are 22 different kinds of strains are there. Each virus has their genetic material is different. All are double-stranded RNA, all are segments. You could see, for example, SIPO virus 1, it forms segments like this. SIPO virus 2, it forms segments like this. If you can see SIPO virus 20, it forms segments like this. So all are double-stranded RNA, segmented genome, but the segment size, their electrophoresis pattern is different. By simply extracting genetic material and see in the database comparing your electroporetic pattern, we will come to know that which virus it belongs. For example, let us see SIPO virus 1 over here. All the SIPO virus are forming same kind of banding pattern. Yes or no? See similar kind of banding pattern. SIPO virus 14, again same. That means that there are 22 different kinds of SIPO virus. All viruses have double-stranded RNA, all are segmented, but only particular group forms particular pattern. All the SIPO virus here we have same pattern, whereas another one different patterns. For example, ours, please go ahead. So this is Muga silkworm, which is infected by this double-strand RNA virus. As you could see, this is the banding pattern. This exactly same banding pattern we can find in the literature of the SIPO virus which is affecting Antheria mylitta, that is tassar silkworm. And exactly again it is showing Antheria proili, oak tassar silkworm. What does it mean this? The virus which is infecting in Muga silkworm, the virus infecting other wild silkworms such as mylitta and tassar is same. They are all having same genetic pattern, right? So go ahead. And again, now you came to know this, all the viruses is same. We want to find based on the RNA banding pattern, we came to know this virus is SIPO virus 4. There is one more method to identify that. I told you that this is a segmented virus. For example, let us see, these are different segments. All segments, for example, this is a DNA RNA molecule. At 5' end, at 3' end, there is a conserved region. Every virus has its own specific unique signatures at 5' and 3' end, right? Every nucleic acid has a definite polarity. 5' is there, 3' is there. So we have to look at what is there, what is the sequence at 5' end, what is the sequence at 3' end. For example, you pay attention on this. This is CPV1. CPV1 at 5' end we have TTA GCC. So if you find TTA GCC in any at 5' end, simply we can call it as this is SIPO virus 1. Why? This particular sequence is unique to this particular virus. Similarly, you see another SIPO virus 22, what is there at 5' end? AT-AGC. This is different. So we want to now investigate what is this regions lying at non-coding regions. What is the conserved regions at 5' end and 3' end? So go ahead. Yes, for that what you have to do, for example, this is RDRP, RNA dependent RNA polymerase. This is a non-structural protein, very much essential for replication of the virus because virus is having RNA molecule now. So now we want to investigate what is the sequence at 5' end and what is the sequence at 3' end. So for that what happened, we have to design different kinds of primers targeting these regions. We will amplify it, you will get bands like this, okay. Then we have to clone and send the vectors for sequencing. Based on the sequence information, go ahead, Please don't make noise, I'll get disturbed, please. Yeah, as you could see, this is the entire sequence of that virus RDRP. Look at this. TA, GA, GC. I told you already, every segment has unique, every virus has a unique conserved sequence at 5' as well as at 3' end. Now we are investigating what is there at the 5' and 3'. So here TAG, AGC. Then we will look into the literature. Which virus is having this particular conserved region? That is nothing but cytovirus 4. So in the transmission electronic microscope as well as scanning electron microscope, we came to know this. This is a occulted virus. The virus is not a rod shape. The virus is a spherical icosahedral. The genetic material is double stranded RNA. However, it is not a single molecule. It is a segmented molecule, and each segment has this particular signature sequence. Based on that, now it is finding that this is a cytovirus 4. So basically we have ICTV, International Committee for Taxonomy of Virus. This is the apex body in the virology in the international level. They actually define the virus based upon this. So as for ICTV taxonomy also, the virus which is affecting Muga silkworm and responsible for huge crop loss is nothing but cytovirus 4. So now first we have identified the pathogen up to this level. Hello, yeah. Then let us see the relationship between cytovirus 4 with other cytoviruses. So that what you do, we have to develop a phylogenetic tree based upon this RDRP molecule. As you could see here, cytovirus 1, all cytovirus 1 form same clade. They infect Bombyx mori, Lemintria disper, Dendrolemus punctatus and Dendrolemus sibericus. These all are lepidopteran insects. These all are actually agriculture pests. Okay, there are 22 different kinds of cytovirus. The cytovirus 1 infect this. Cytovirus 4 has so many insects. It is going to affect. So that's why all 4 will form in one clade, phylogenetically, because during the evolution all have come from the same ancestors. That's why in the same clade we will have all the things. So look at here, cytovirus 4. It is forming this triangle, as you could see, triangle. This is cytovirus 4, which is affecting Muga silkworm. Is have 100% similarity with Antheria mylitta. That's why in our genetic RNA pattern, they are having similar kind of segments, similar kind of electroparogram, right? So based on this, we'll come to know this. Now it's very clear. Cytovirus 4, which is infecting Antheria assamensis and mylitta, is similarly related. Further, this cytovirus 4 has close relationship with cytovirus 22. That means cytovirus 4 and 22 has something is, something is there in the evolution. Both have come together, right? So such type of information we can run. Then this is a kind of heat map, because while you are drawing such type of a phylogenetic tree, we are having RDRP sequence, and we align RDRP sequence, we find how they are similar or dissimilar in nature. Then we can see such type of structures. We are calling it as famously heat maps, where the blue color indicates that all are same. Yellow color indicates that they are distant from each other. For example, let us see here, cytovirus 5 this is cytovirus 7, and this is something around cytovirus 50. So you can see more yellow color in this particular isolate. That means that among all these 22 isolates, these 3 particular viruses are very much unique. They are not at all showing any relatedness with another cytoviruses. That means we need to investigate their evolutionary pattern as well as their etiological studies. Yeah. Then, so now we have identified the pathogen as to be cytovirus 4. So similarly, during this protocol, we have developed a protocol. For example, if any scientist to work on cytovirus, or you want to know which type of cytovirus he is handling with, so we have developed a protocol like this. Whatever you have seen, first extract the polyhedral bodies, see under the microscope, any polyhedral bodies are there or not. If polyhedral bodies are there, then extract the genetic material. And you have to find whether it is a DNA and RNA. If it is RNA, then again we have to develop electroparogram. And we have to compare this electroparogram with the existing database. Parallelly, we have to identify the conserved region, signature sequences at 3' and 5' end. Okay, then you develop a phylogenetic tree. All together, finally, if you can generate information, all this thing, finally we can identify the virus at strain level. Please go ahead. Now, the first step is identification of the pathogen. The second step is to develop diagnostic methodology. we have developed a multiplex PCR. What happened? These wild silkworms are susceptible to not only viruses. Along with the viruses, there are other diseases. For example, let us see here. There is one virus, Ifla virus. There is febrin. This is one pathogen. This is also one pathogen. The third one is viruses. Generally, if you want to identify virus, we do PCR. At a particular time, you can identify only one particular type of pathogen. However, here we can develop a technique known as multiplex PCR. All the pathogens can be detected in a single PCR reaction. So that type of assay, because what happened at the farmer field, we want to know which type of pathogens are there. By simply doing one simple technique, we will identify what type of pathogens are there. So the first one is to identify the pathogen. Second one is to develop a diagnostic methodology. The third one is to develop control measures. Then only farmer will get benefited, right? Go ahead. Yeah, please. The same technique we have used in the field samples. See, as you could see, in some fields we are having 3 bands. That means that particular rearing field, that particular location is having 3 different type of silkworm pathogens. Then immediately we will start control measures. Go ahead. This is another technique to develop a diagnostic methodology. You all know how COVID is diagnosed. Which techniques? COVID. What do you do? RT-PCR and then? One is RT-PCR. RT-PCR, the first methodology, what is there? Rapid antigen test. You know, one small strip will be there. They take swab sample and simply they will add and see the bands. So what they do actually, that is known as rapid antigen test. That particular strip is actually coated by antibodies specific to pathogen proteins, right? Then immediately we can see the bands. If bands are there, positive for COVID, and add on to this stripe and see whether any bands are there. So this is just a trial for developing diagnostic model. Yeah. Then again, in the northeastern region, farmers used to rear Moga silkworm. Parallelly, they can also rear Aedes silkworms. We want to know, so this pathogen is hypovirus 4, whether this particular virus can infect other silkworms or not. That is known as cross transmission, whether cross transmission is there or not. If the cross transmission is there, then entire sericulture industry will collapse, because this virus is affecting Moga silkworm and we are witnessing 80% crop loss. In the same region, there is a huge airy crop is going on. We are very much interested now whether the same virus can able to infect the airy silkworms or not. For that we have to do study. And since it is RNA virus, it's a little tricky. Always extract the RNA, we have to develop cDNA, then we have to do RT-PCR. Whatever bands we have seen, no, all are RT-PCR based bands. Yeah, as you could see here. So these are the airy silkworm larvae. We fed this larva with virus specific to only the wild silkworms. So when you are talking about wild silkworm, these are all lepidopterans. Go ahead. Then we are interested to see whether lepidopteran and other insects are susceptible or not. This is a Bihar airy caterpillar, one of the agriculture pests. We have found that this virus is unable to infect this. Go ahead. This is a semi-looper, one of the pests in castor. Okay. There is also, we could not find the expected band here. It is not at all affecting, although it is a lepidopteran insect. Then Spodoptera. Spodoptera is one of the famous agriculture pests. However, this is also free from that virus. So with this etiological studies, we could be able to see this. This virus is highly specific to Vanya silkworms, which does not affect another lepidopteran. Yeah. Then, so we have identified the virus. developed certain diagnostic methodology. We have studied cross-transmissibility. Then we could able to develop certain control measures. So for that what happened? We have selected these chemicals and we have applied on this particular virus. Go ahead. The results indicated that slack lime, that is calcium hydroxide at 2% and bleaching powder at 2%. If you can mix it, it is more effective. That means that if you have a virus crop loss in the rearing field, we have to apply this particular reagent in order to save the crop, right? This is known as control measures. Okay, so we simply apply these chemicals on the polyhedral bodies, whether the polyhedral bodies are able to dissolve or not. Once you are able to get the dissolution of all polyhedral bodies, we can recommend the farmers, you go through it this type of protocols. Then only they will able to sustain in the field. Yeah. Similarly, am I going fast or what? I will ask you 2 or 3 questions, okay? You have to tell. So please pay attention. This is another insect that is known as Antheria myelata, tassar silkworm. In the tassar silkworm, these are the few of the symptoms, virosis symptoms. As in Muga, earlier we were not knowing this, which virus is causing this type of crop loss. Okay, please go ahead. While we were searching in the wild silk base, this is a database where you could get lot of sequencing information of wild silkworms. Fortunately, we found a signature sequence. This is actually a transcriptome, RNA level sequence of the tassar moth. This is actually not a host. This is a virus sequence. How come a virus sequence came into host sequence? That is our interest. So then immediately we have designed primers. Please go ahead. Then we have developed a PCR protocol assay. What is this virus? This virus is known as IFLA virus. This is single stranded RNA virus. The earlier one is hypovirus, that is double stranded RNA virus. This is a single stranded RNA virus. Then immediately we have actually deduced the genetic entire genome of the virus. The entire genome is 5' and 3' end. It's around 9,700 base pair. The entire virus genome is this. We have again 5' and 3' end. At 5' end we have structural proteins. VP1, VP2, VP3 like this. At 3' end we have RDRP. So, established the genome organization of the Antheria myelifla virus. Please go ahead. Then again, as you could see here, this is actually a phylogenetic tree. Again, it is showing Antheria ifla virus. That means that the ifla virus, which is affecting myelita and prioli, are both are same. It is further confirmed with the different type of tissues. The virus is present in all parts of tassar silkworms. As you could see, fat body, midgut, silk gland and all. These are different body parts of insects. All body parts we can find. That means that this is a systemic infection. There are 2 different kinds of viral infections. One is localized, the other one is systemic. Systemic means you can able to find the virus signature throughout the insect body. Localized means only in one particular tissue only we can be able to see. So this particular one, sorry. Then we have done in which body parts this virus will be more, like where it exactly multiplies more. So for that we need to have a quantitative real-time PCR. As you could see here, the fat body and silk gland, we have more multiplication of this virus. The same virus we have find in another silkworm, Antheria prioli. But interestingly, in Antheria prioli, the same virus is present along with another virus. That means there are 2 different kinds of virus we can find at a time. That is known as latent infection. So one is ifla virus IV, then is NPV. Yeah, thank you. This is my team. He is our HOD, head of the department, Dr. Ponnuvelu sir. This is my team. These 2 are bioinformaticians. We have very good infrastructure to do bioinformatic analysis. We have power stations and servers are there. So my collaborator in Northeastern region, this one, Indir Kumar is a Tamilian guy, and these two are also working in the Northeast because our institute is located at Bengaluru. We don't have Moga silkworms here. So if you want to work with them, so we have to make collaboration with other scientists in the Northeastern region. And she is my student, she is doing PhD in this. The second part of work, especially IFLA virus, this work is done by my in charge, Ponnivelu sir, with collaboration with a Swedish counterpart. He is only 10 years from Swedish Agriculture University, Uppasala. So there are certain techniques we can't do by ably, collaboration is very much required. So we are into this only 10 years from Swedish University, we work together. I would like to express my sincere thanks to Neethrajan sir and also Shobhana Ponnivelu madam for giving me this opportunity to share few of my research findings with students. Thank you.
Our single stranded RNA, which one is the highly virulent virus, sir? Single stranded DNA or single stranded RNA, which is the highly virulent virus?
DNA, sir. Single stranded DNA is the most virulent. Single stranded RNA will give you a chronic infection. Immediately silkworms may not die. Okay, it takes so much time actually. So, but single stranded DNA is
and other tests. Only one protein test was only done. So you have just seen whether the protein is there or not. So as of I can see, all this virus, some of the viruses has no structure at all. So what do you think of that perspective?
No, madam. Cypovirus, actually, we have identified this pathogen is a cypovirus, madam. Cypovirus has well-defined structures are there. It is actually spina rheoviridae. It has a spike proteins, which is caused by segment 1 and segment 2, actually. So here, in order to develop a diagnostic methodology, only polyhedron we have cloned. So then what happened? We have sent for our genome for sequencing. Once you get entire genome, then immediately we will come to know the proteins and all, madam.
Thank you, sir. Thank
you. Good afternoon, sir. I have one doubt that only virus will affect the silkworms or a virus affecting bacteriophages there, no, sir? Like the bacteria will also cause any viral disease in silkworms, sir?
No, virus, each virus has a specific host. So this virus is affecting silkworms, right? So bacteriophages, matlab, they are the viruses which affects only the bacteria, not the silkworms. So bacteriophages, the host agent is different. That is bacteria, okay? They cannot infect silkworms.
Only virus will affect or any other parasites?
Yes. Yeah, I will tell you. Generally, we have 4 different kinds of diseases we can find in sericulture. So one is virus. Okay, we have seen all these things. Then bacteria. We can Bacillus thuringiensis, Streptococcus species, some Bacillus ameloleukophaciens, and Serratia marescens. Okay, and Klebsiella, Enterococcus. These are different kinds of pathogenic bacteria. This is bacterial disease. The first one is virus disease. The third one is fungi, muscardine. So you may be knowing, entomologist, muscardine is caused by different kind of Beauveria bassiana or Metarhizium species. And the fourth one is Nosema species, that is caused by, the disease is pebrine. Nosema is pathogen, bacteria is pathogen, fungus, muscardine is a pathogen, and virus is the pathogen.
Yes, sir. Thank you, sir.
Good afternoon, sir. Sir, like BT cotton, can we able to produce any variants in mulberry leaves like that can kill the viruses?
To kill the virus, see, generally, yeah, this is RNA, right? In order to break down this RNA, we have to silence that particular RNA molecules. So we are doing what basically there is siRNA, miRNA. These are the small RNA molecules. Will go and bind your targeted virus and they will degrade. So we can use transkingdomal approach. So we can express those miRNA, a small iRNA into this silkworm. Then only we will be able to save the virus. Sometimes what happen? See, I'll tell you. There are 2 silkworms. One silkworm is susceptible to the virus, but the other silkworm is not susceptible. That means the genetic content matters, right? So first we segregate which are resistant to this virus, right? So for example, we have collected 4 silkworms. All 4 are resistant to the virus. Then we make a breeding approach. So we want to develop virus resistant breed. So that type of work is also we are doing that.
Thank you, sir.
Sir, this is a very general question, and I know we earn a lot of money from it's a very commercial product, but why are we torturing those small insects? Don't we have other means of producing silks other than only the worms, relying on the worms and killing them? Is that the only way, or are there other means? Because we have developed technically very far, no?
Madam, this is very nice question. Actually, what happened? Nowadays, Central Silk Board is also concerned a lot about this. Now we are coming up with a technique known as Ahimsa silk. So Mahatma Gandhi is our role model. So what is meant by Ahimsa? Basically, the cocoon we have seen, no? The second silkworm, eri. So no need to cook those cocoons. We cut the cocoons, we remove the pupae, whatever the cut cocoons, we spun the silk and we do that. So, no, the pupae will be useful for seed generation. We will use this pupae, we will keep it like this. After 10 or 15 days, we will get a silk moth. So they couple each other and produce eggs. These eggs we will sell. So the complete life cycle we will follow. Especially, madam, Jain people and Buddhism, they won't wear silk. They think that silk is coming because of killing the silkworms like this. So that's why now we have started that, madam.
We hope that you had a wonderful time in this DBT sponsored first international conference on the significance of gain and loss of function and impairing transcription factors causing genetic disorders in humans, and enjoyed our hospitality too. Setting goals is the first step in turning the invisible to visible, says Tony Robinson. Our goal is to attain a prosperous valedictory function. We humbly believe that we achieved our objective and as well as the goal of our conference. Yes, ladies and gentlemen, we are at the brink of this international conference. This gathering is reconvened to witness the auspicious valedictory function. Pleasantries. Honoring the guests always the traditional practice of our culture system. We are happy and humble to recognize and honor the guest on behalf of our management, for which I am delighted to offer a happy welcome to and as well as the invitation to our Principal Sir, A. Muttumani, to honor our chief guest of the valedictory function, Dr. Gangavarappu Subramaniam Sir. Thank you, sir. This culture system is known for its reception. We Indians greet all with our smiling faces, knowing that all the humans are nothing but the embodiments of divinity. To welcome this august gathering, I kindly request Dr. G. Kirtana, member of the organizing committee of this conference, to propose the welcome address.
Thank you, sir. Yeah. A warm and a cheerful noon to everyone present here. We are pleased to welcome everyone for the valedictory function of DBT sponsored international conference on the significance of gain or loss of function and impairing transcription factors causing genetic disorders in human. With immense pleasure, we welcome our dearest principal, sir, Professor Dr. Muttumani, sir. Welcome, sir. Distinguished chief guest, Dr. G. Subramaniam, sir. Welcome, sir. We render a warmest welcome to respected head and faculties of various departments, distinguished researchers, participants from all over the world, dear colleagues, and dear my students. I welcome you all on behalf of PG and Research Center in Biotechnology. Welcome, everyone. Yeah, and my heartiest welcome to our eminent speakers, Dr. P. Sundaresan, sir, Dr. G. Muthuswamy, sir, Professor J. Martin Collinson, sir, Dr. Akshaya Murugesan, Dr. Arjun Masurkar, Dr. Ruby Vijaykumar, Dr. Satish Kumar, and Dr. G. Subramaniam, sir, who had shared their invaluable time and knowledge with us. I welcome all the guests from different colleges and universities from different parts of the world. We hope that the two-day conference would have helped the participants to expand their mental horizon and enrich them with knowledge from all around the world. We welcome our media friends who are very supportive these 2 days. Welcome media friends, and once again, we welcome you all for the valedictory function. Thank you.
Thank you, ma'am, for a nano-sized welcome address. I kindly invite Dr. P.S. Shobna for a pleasant task.
Hello, everyone. I'm here to introduce our chief guest, Dr. G. Subramaniam. He's a scientist C from Central Silk Board, Bangalore, Karnataka. He has done his post-graduation in microbiology in College of Science and Technology, Andhra Pradesh. He has done his PhD in Department of Microbiology and Biotechnology in the Maharaja Sarojirao University of Baroda. He has served as a principal investigator in 4 projects and co-investigator in 6 projects. He has also won several international and national awards. He has presented several international and national seminars, workshops, training courses. And here, thank you for coming, sir. I thank Principal sir and HOD sir for this opportunity. Thank you, sir.
Thank you, ma'am. The interaction of knowledge and skills with experience is the key to learning, says John Dewey. From the bottom of my heart, I render my cheerful welcome to this experienced stalwart, man with voluminous experience, chief guest of this valedictory function, Dr. Gangavarapu Subramaniam, sir, Scientist C, Seri Biotech Research Laboratory, Central Silk Board, Government of India, for his valedictory address. Sir.
G. Nithirajan, sir, Head of the Department Biotechnology, and also convener and organizing secretary for this 2 days international program. Dignitaries on the dais and off the dais, dear student friends and participants, a very good afternoon. Yes, a very good afternoon, one and all. Yeah, we had already interactions in my deliberation. However, I have few points. The first one, we know this cell. What is cell? Cell is a structural and fundamental unit of a life. In mammalian species, we have approximately 200 different kinds of cells. These all cells are highly heterogeneous in nature. Their structure, their shape, as well as their functions are very unique. What is the driving force which actually conferring this particular type of special phenomena? So that is nothing but this transcription, initiation factors. Transcription initiation factors actually decides which type of proteins has to express or it has to be repressed. They do a lot of moral policing in our body. So obviously, in the first day, Sundaresan sir was telling, he was pointing out central dogma of life, DNA to RNA, RNA to protein. So RNA to protein is one of the vital step in our body, in our cell. So that is nothing but a protein synthesis. But this protein synthesis is highly regulated phenomenon. It's tightly regulated by transcription factors. Since I must tell you, everyone knows what is the size of human genome. If you can anyone tell, what is the size of human genome? Anyone? Please raise your hand. Should know, students know. Biotechnology. Yeah. Sorry. It's around 3 billion base pairs. Though it's a vast genome, we have 20,000 different kinds of protein in our body. 20,000 proteins. Among these 20,000 proteins, 2,000 proteins are coded by particular genes that is known as initiation factors, transcription initiation factors. So total proteins are 20,000, 2,000 are your transcription factors. So that means 10% of our human genome is actually coding transcription initiation factors. So you should realize the importance of this transcription initiation factor. On the other day, I'm new to this subject basically, I'm a virologist. And the other day I was actually reading few of the articles. What happens if any, find mutations or any defects in this particular transcription factors. So it's very surprising to see this. Almost all the chronic inflammation, whatever we face now, a post-COVID scenario, all is due to this post-transcription factors. So asthma, it's responsible by transcription factors. Different kinds of cardio, diseases, again, same. Defects are nonsense or missense mutations in transcription factors. And again, we have seen a lot of NVIDIA. So I must congratulate Nithirajan sir. He has done a substantial research, especially this PAX6 and FOXA, something like this. He has done a lot of work how this transcription factors is actually involving in NVIDIA, congenital NVIDIA, means eye development and eye disorders, retina manifestations and all. So your eye is deciding by transcription factors. Brain is actually controlled by transcription factors. Heart, liver, in whatever disease you name it, largely or to some extent is actually influenced by the functioning of transcription factors. If the genes are very good, there is no mutations, everything will be fine. There could be a good equilibrium. Homeostasis will be there. But if anything is defected in the transcription initiation factors, obviously we will end up with chronic or maybe acute diseases. So I think that's the reason he has selected this topic. And this is a highly advanced and very specialized topic. For selecting such a wonderful, very much required in the hour. So I must congratulate the college as well as the principal, sir. However, nevertheless, to do such type of studies, we need to have, first of all, finance, funding, and also good infrastructure. Most importantly, we need to have collaboration out of borders. Okay, then we can do very. So I thank the principal sir, Nithirajan sir, and Sobna madam for giving me the opportunity. And I request students. So science is such a vibrant, field. Should have very constructive and positive mind, and kindly continue your career in the scientific field. You are the future of our nation. That's my request. Thank you, one and again. Jai Bharat.
Thank you very much, sir, for the invitation, and surely we will sing to others is an art of flowering your heart. Presentation or oral presentation, best 3 oral presentation are identified and recognized with a shield and certificates. To distribute the certificates to the winners and the participants, I feel honored in offering a cheerful welcome to the dignitaries on the days to come forward to distribute the certificates. Best 3 oral presentation. The 3rd prize goes to Mr. Tarun Kumar and Kiran Kumar, B.Tech Biotechnology, Adiyaman College of Engineering. Second prize goes to Ms. C. Divya from Periyar University, Salem. Second prize, C. Divya from Periyar University, Salem. And the first prize goes to Dr. Lokesh ST from Kuvembu University, Bengaluru. For getting the certificates from college-wise, I read out a representation from each department. Kindly come and collect. Ms. Mary Blessy, Department of Biotechnology, Adiyaman College of Engineering. Followed by Mr. Tirunavukarasu, Department of Chemical Engineering, Adiyaman College of Engineering. Mr. Arul Kumar K. from Periyar University, Salem. Arul Kumar K. It's not there. Okay. Arun R. from 3rd BSc Microbiology, MGR College. Arjun C. 2nd BSc Microbiology, MGR College. Archana S. from 3rd BSc Biochemistry. Biochemistry of MGR College. Any representation from Biochemistry? No. Okay. Yes. Come first, please. Settle down. Kirtika GV, first BSc Chemistry, MGR. Thank you, sir. I read out the colleges who are going to get the certificate in absentia. St. Joseph College of Arts and Science for Women, Hosur. Aringaranna College of Arts and Science, Krishnagiri. Sona College of Arts and Science, Salem. Batmavani Arts and Science College for Women, Salem. Mutayamal College of Arts and Science, Rasipuram. Christ University, Bengaluru. The Oxford College of Science, Bengaluru. Jain University, Bengaluru. Christ Jain College, Bengaluru. Reva University, Bengaluru. AMC College of Bengaluru. An appeal to all the winners. Happiness is like a perfume. You can't spread on others without getting a few drops on it. So always be happy to make others happy and make your knowledge infectious for the betterment of the society. Opportunities are equal for all, but feedback? No. Gratitude is not only the greatest virtues, but the parent of all. To record our gratefulness, I would like to invite Dr. R. Muthuswamy, organizing committee member of this conference, to propose the vote of thanks.
Very good evening to all. I deem it a great honor and a privilege to propose the vote of thanks on these memorable occasions. So let me first of all start by giving glory to the Amity Good for making today occasion a resounding success. So I would like to thank our chief guest, Dr. Akshaya Murugesan, and then Arun V. Murugesan sir, and then Ruby Vijayakumar ma'am, Sadish Kumar Rajashekaran sir, and Sundaresan sir, and Govardhan Muthuswamy sir, and Dr. J. Murugan sir, and Govardhan Muthuswamy. And also, I would like to thank Dr. Gangarajupu Subramaniam sir for his wonderful presentations. My heartfelt thanks to all the head of the departments, various departments, and the faculty members, students from various departments, and also my special thanks to our own departments, biotech department students. So without their contributions, we cannot finish this conference, other supports during the preparations and arrangements all. Especially my sincere thanks to third years and even second years, all the students And also I grateful to thank our management and other non-teaching staff members. And finally I would like to thank our principal, honorable principal, and then our organizing committee members, Needhi Rajan sirs, and the media peoples. So without their support, so we cannot export these conference throughout the world or everywhere. So I finally thank our media peoples and also some of the support members, for example, for giving the current supply, everything. That means for non-teaching staffs. So thank you for giving this opportunity. Thank you one and all. Thank you.
भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्छल जलधि तरंगा तव शुभ नामे जाहे तव शुभाशिष माहे गाहे तव जय गादा जनगण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Life is about the people you meet and the things you create with them. Hope we will meet you once again in second international conference with us next year. Until then, ma salama from the team. It means goodbye until we meet again in Arabic. Thank you very much. Thank you.